Welcome to the Ecology Center seminar today. Our guest today is Dr. Moises Exposito Alonso. I hope I pronounced that correctly, known to his colleagues as Moy. Moy is an evolutionary geneticist with the Carnegie Institute for Science and an assistant professor in biology at Stanford. He earned his PhD in ecology, development, genetics, and evolution. That's quite a lot of things all at once at the Max Planck Institute in Germany. His Master of Science in quantitative genetics and genome analysis at the University of Edinburgh, and his bachelor's in ecology and evolution at the University of Seville, Spain. So he is truly an international scholar with a range of prestigious awards throughout his very young career. His research is currently funded through several grants from USDA, Carnegie, NIH, and DOE. So much of his work to date has been using Arabidopsis thaliana, which is a model species for adaptation and evolution. And he's also been branching out into some non-model systems, according to his CV, including phytoplankton, corals, Joshua trees, pennycress, and even aspen, which makes me very happy. Overall, Moy uses large-scale ecological and genomic sequencing approaches to help determine whether and how plants will evolve in response to climate change. And of course, those are questions that we're all very interested in. So now, if you would, everybody would turn on your mics just for a second. So Moy knows that we are real people, and let's make some noise welcoming Moy today. So, okay, now turn it back off.
So I think that's my cue to start. Thank you, Karen, so much for the introduction. And that was actually a pretty good idea to make some noise at the beginning of the seminar and get that spirit up and some adrenaline to start talking science. So it's a pleasure to be, at least virtually, giving a talk at the Ecology Center. And so today might be kind of an edge type of talk for you guys, as the majority of our research deals with genomics. But I will tell you how we are trying to use this genomics and evolutionary thinking to address climate change ecology questions. So the model system, or the question, is really motivated by our impacts in the world. And these are the two main impacts, I would say, that we are posing on ecosystems. The first is land transformation, direct removal of primary habitats into human habitat. The other, which is expected to be actually overpassing land use transformation in mid-21st century, is the effects of changing climates that we've also created. And some of those effects are pretty clearly observable already. For example, in the case where tree lines are moving and advancing to higher altitudes. And these changes in space of where species can live are increasing, among other things, the extinction risk of many species in the world. How people have typically approach this question of studying these two impacts, main impacts of human on species, is through the study of where each species is, right? Their geographic distributions. Because we have pretty good records since the naturalist expeditions a few centuries ago of where biodiversity is. And then you can pose the two scenarios of if there's a climate change, and other impacts too, but if there's a climate change, one of the options for species is to track where the optimal climate is, for example, by migrating in a northern temperate species polewards to the North Pole. The other, if species do not actually have the ability to disperse so fast as it appears to be in many species, and it will be expected, for example, of plants. Species might be losing populations in the fronts where new environments are encountered and where the species is not able to survive anymore. And this really was kind of the basis for many species distribution analysis and other type of spatial analysis to predict how many species might get completely shrinked in the 21st century and therefore become extinct. And this is about 3 to 25%, varying a lot depending on the methods, assumptions used, as these are predictions to the future. But what about adaptation? One would argue that if populations were able to adapt to the new environments, that will preserve where they're found, but that is rarely quantified because it requires much more in-depth knowledge of a species. Now it's been recognized that this third option is very important to maintain. And the fuel for that adaptation is the variability within species, genetic diversity. And as of July 2021, it's surprising how long it took, the United Nations Sustainable Development Framework now includes a main statement to protect 90% of genetic diversity within all the species that we want to protect, which are all of them, which is a very bold aim, but it's important because it recognizes that dimension of biodiversity and the importance to maintaining evolutionary potential of species. So in order to do this, we need to start thinking of this framework of predicting biodiversity changes or extinction risk instead of at the species level, at the genotype level. Right, so we might, instead of think of a distribution of a species, we might think of distributions of specific genotypes, each of which might be adapted actually to different environments where a species is found. And when impacts or climate change come, we can start making genotype-specific hypotheses. For example, there might be locally adapted genotypes in some areas of the distribution that might have certain ecological strategies worth understanding. We might try to look at how far gene flow has to be, or what are the gene flow connections between populations in order for locally adapted genotypes rescue populations that lack them. And we need to understand also how species are losing areas where they can live due to human transformations, and therefore genotypes are being lost. And I argue that now we have enough data for many species to start asking these questions at a scale. And these are just some of the species that our lab has been recently looking at. Although our model experimental system is Arabidopsis thaliana, which you might have heard of it as the model plant for geneticists, molecular biologists to study specific genes. But in the last decade or two, it has really transformed also into a model for eco-evolutionary research. And I think it's perfect for some reasons that I'll show you to study climate change adaptation. Some of the features of Arabidopsis that will be helpful to understand certain analysis later on is that Arabidopsis is an annual plant. So it germinates in autumn, overwinters, and reproduces and dies in the spring, and then seeds stay as dormant over the summer. It has been seen in many extreme environments. These are two pictures that I took of two flowering Arabidopsis, one in the snow, one in a sand dune. And it has a very broad geographic distribution. So it's native to Western Eurasia, and the warm edge is in North Africa, but it's also dispersed to other areas in the world recently. In 2016, most importantly, the first over 1,000 complete genomes were published, or we published this resource, and with a seed bank or seed germplasm associated to conduct experiments. All right, so that's kind of the background of Arabidopsis and our framework. And today I'm going to ask 3 questions. Hopefully I have time to go through all of them. Otherwise, we're going to skip the third one. In the first, I'm going to ask, how are these genotypes distributed in space, right? That is one fundamental question. And a follow-up on that is how those genotypes might be lost with human impacts. Then I will ask, well, is this differentiation between population, genetic variation, really increasing the potential to adapt to climate extremes? So we'll ask several related questions, such as where those important genotypes are, how many are there, are selective pressures, that is, what genotypes are selected where, predictable, etc. And finally, we'll ask a new question of work that is unpublished yet, asking whether even if populations have genetic variability, how fast this can happen, right? Because if it doesn't happen fast enough, they will not manage to persist. So let's get to it. In the first, it's a project really less than 1 year old in the making, despite I've been brewing it for a while in my head. It was to ask about where genotypes are within species distributions and how the loss of area is going to affect the loss of genotypes. And I tackled this actually being inspired by models that were used to predict how many species are at risk of extinction. So this is based on that the majority of species are rare, while some of them are common. And you might think of an oak forest, that is the common species, but then you find many, many species that are rare. Based on this, well, this is something that has fascinated many ecologists, and there's been many proposals of functions on how this should work. But one of the consequences of this commonness of rarity of species is that somehow ecosystems that have larger areas, for example, islands of Galapagos, and this is one of the first data sets that was shown or was used to prove this theory, those islands that have higher areas, they have a higher capacity for harboring species. And this was proposed to follow a power law that it has the form of number of species is equal to a constant times area scaled to a scaling factor Z. This will become evident why it's interesting in a second. This feat was not amazing in one of the first papers, but it has been thoroughly tested and is around 30%, meaning if you increase 1 order of magnitude the area of an ecosystem or sample 1 order of magnitude more, you see on average 30% species more, and that's larger ecosystem. So in the first assessments of species predictions or species loss under climate change, this relationship was used with the idea that if we have the whole Earth as the total area, and we are reducing X% of the area available for native ecosystems, natural ecosystems, we will then, with a back of the envelope, predict how many species cannot harbor the Earth with less area. So this, when I was reading about all this, I couldn't stop by observing many similarities to mutations or alleles. And I will be using mutations, alleles, and genotypes kind of indistinctively. And you can ask me if there's any confusion there. But we think of essentially like a letter ACTG that is different in a new offspring that has occurred just due to random DNA copy error. So mutations in space might also be distributed like species, like some Gaussian distributions. And we might ask the question, if we start sampling more and more areas of a species, how many more mutations do we discover? How many more genotypes we discover? So I used the Arabidopsis dataset that covers a very large continental fraction of Eurasia, and started sampling areas of small or large sizes. And I did this with a number of statistical approaches like bootstrapping and so on. And then directly in silico, in my computer, counted how many mutations I would observe in the genomes within those areas. And then I plotted it here in a log-log scale. To my surprise and excitement, this followed also something similar to a power law distribution, which is the number of genotypes is equal to or is proportional to the area sampled scaled to some coefficient. And that coefficient seemed to be very similar to the species area relationship. So essentially, this tells us a species, or if we sample a species 1 order of magnitude more, again, we gain 30% more mutations. So we tried to derive this from population genetic theory, and actually it makes sense. And it's just a result of a balance between the generation of new mutations and properties of a species based on how much they can disperse and how much gene flow. And of course, there are multiple relationships depending on how fast individuals within a species can move. And there we established 2 boundary conditions. So one of the conditions is a situation in which a species is completely panmictic. Any individual can move essentially infinite in space. So that will be just a sampling individuals within a population that are all well connected. What that creates is that in space, as you sample more area, you might encounter a mutation that you had already sampled here, so you don't double count it. And this kind of saturates, so it has kind of a very shallow slope, right? Because once you've discovered the majority of common mutations, even if you sample more area, you've already discovered the majority of the genetic distribution, of the genetic variation. However, when populations are disconnected from one another, the species genealogy is kind of structured in space, right? So we have this, what is called coalescent tree, like a genealogy, where individuals from here do never reach this other side, right? So you might expect that the mutations that are encountered in this side of the distribution are not the same ones as in the other side of the distribution. And what this causes is a power law to emerge. And this in the most extreme scenario where each individual of the, or each population of the species has unique mutations, the limits will be z equal 1. But intermediate gene flow will be about 0.3, which is what we had found with Arabidopsis thaliana. All right, so this was very exciting. But then we thought, can we use this to understand what might happen if we start cutting the forest of Arabidopsis thaliana of the world? So we started simulating within the Eurasian distribution of Arabidopsis thaliana different scenarios on how humans might impact the environment, might remove ecosystems of Arabidopsis thaliana, like randomly, or some radial extinction, or some warm edge extinction. Many people believe that in the warm edge conditions will be too hard for a species to survive in the future. They've never encountered them, so they will be the first to be lost. So I did this in the computer, keeping track on how much area has been lost for the species and counting the mutations. And then using the power law that we had discovered, projected that theoretical scenarios as well as the empirical simulations. And what this showed is that there is a pretty good fit. So it seems that as species ranges are lost, or as species distributions are lost, genetic diversity lags behind a little bit until it finally collapses. And this is something that has also been proposed for the species numbers within an ecosystem. So this was very exciting, but it wouldn't be very appealing if this was just an Arabidopsis specific situation, because it has lots of genomes and it's very broadly distributed. So maybe it's kind of a weird species. So we downloaded large genomic data sets of 19 other species harboring or creating a data set that was humongous of 200 million mutations, and then repeated this for each species. And unsurprisingly, most of them followed some type of power law with slightly different coefficients, but this generally means that most species might lose genetic diversity kind of at the same, more or less with the same, following the same dynamics. So this could help us extrapolate and see how far we are from the United Nations proposal of keeping 90% of all genetic diversity in the world. So now this will be very complicated. We will need to get information of each species, really get, for example, from the IUCN, how much area has been lost in the past, etc., and make a number of assumptions. But we just thought, let's go simpler, do a back of the envelope calculation just to see if we can get a first order estimate. So using remote sensing information and published reports on how much fraction of the Earth we've already transformed, we found that it was always about 40%. And you can see in the back, loss of forest tracked from Global Forest Watch. And we thought, okay, if we use the coefficient that we've estimated to be an average for across species and this minus 40% area reduction of the world, what might be the loss of genetic diversity? And this, on average, was something more than 10%, suggesting that we might be already behind the proposal from the United Nations. Now, there's going to be lots of noise in this estimate, and it's something that we're actively working right now, and we're happy to receive feedback. We put a preprint on it a few months ago, but instead of just proposing the average genetic diversity loss is about 10%, we thought, let's plot all the scenarios. So all the scenarios really are, what are all the percentages possible of species losses, of species area losses, right? It can be between 0 and 100%. What are all the possible distributions of mutations area relationships from 0 to 1? We derive that also mathematically. What are the estimates that we found per species here in salmon color? We plotted them. And what are the estimates from a number of global reports on habitat transformation? All of those. If we intersect them, they are around 10%, as we were saying, with a pretty broad confidence interval. But this kind of scenario mapping is, I think, something that we can develop and use practically as we get better and better information on mutation area relationships per species, as well as impacts per species. All right, so the first question we answered, we can model the spatial process of genetic diversity loss using a power law, and this suggests that we might have already incurred in pretty large genetic diversity losses, as we would expect logically by a transformation of 40% of Earth's ecosystems. Now, does that loss of genetic diversity really matter? So that's the second question. Can we use Arabidopsis thaliana to understand or to guess what might be the potential of having genetic variation to adapt to climate extremes? Okay, so in order to try to think globally how we can predict what might be the adaptive potential or vulnerability of different populations to future climates, I think we need to understand a number of things or properties of a species spatially. And one of them is start making geographic maps of where potentially adaptive alleles that confer some increased fitness or increased adaptation are. For example, and I'm presenting this kind of as a cartoon with kind of a probability, right, along this space. There's some feedback, right? The other thing that we need to know is where are those potential alleles adaptive? our assumption nowadays is that the alleles that are selected at each location or that are adaptive in each environment where a species lives, those are where they are going to be. For example, we can think of an allele that might increase some type of drought adaptation might be found in a region of a species where there is droughts, but in a region where there's high rainfall, that allele might be negatively selected due to some trade-off. We could put forward that hypothesis, and I'll show you some evidence for that. Now we need to understand this map of adaptiveness of an allele and how it is going to change with climate change. And we can use present and future climate predictions to try to estimate that change. And finally, we need to integrate these two, what genotypes are present locally and how their selection or their adaptiveness is going to change to the future to make some map of vulnerability, right? Mismatch between the perfect genetic makeup of a population and the environment that is going to be present in the future. All right, so let's see what we can learn about those different parts. How do we identify genetic variants that might be adaptive in different environments? To do that, we have to do experiments with genotypes, for example, 500 genotypes from the Arabidopsis 1001 collection in different locations. So this is a picture of a common garden where I planted this over 500 genotypes in Spain. Once we grow them there, we can measure a number of traits, for example, survival, and you will see that the differences between genotypes of the same species are pretty dramatic. These are pots at the same water content level. And combining genome variation, that is specifically what are all the mutations of these ecotypes, and comparing those ecotypes with the ones that didn't survive, we can make statistics to try to understand what mutations are enriched. And this is called genome-wide association approaches. And we can plot the significance of that association, now minus log 10 p-value, to see very, very low p-values for all the SNPs, all the positions in the genome for which there is two alternative alleles or two alternative mutations. And those are the ones that we see here. This is called a Manhattan plot. What this allows us is to visualize areas that are particularly enriched for genotypes that explain the survival of certain plants. Some of the take-homes that we found doing this for multiple phenotypes, such as survival under these common garden experiments, survival under drought conditions, and a number of other phenotypes, is that generally we find a very polygenic architecture. That is, it's not only a single region in the genome where there is a specific variant of a gene that explains that a plant survives. And this might be in contrast to a number of cases in disease resistant, where there's typically like a gene-to-gene relationship. And you can imagine that a plant that manages to survive such harsh conditions will have to do some type of coordinated effort in multiple traits, such as roots, stomatal control, growth, etc. And this is what we find. All right, so some of the once we identify some of these potentially adaptive alleles, one of our questions is locating them geographically. So adapting species distribution modeling, but to mutations, we can create probability models of multiple of those alleles that we've identified in previous experiments. So combining the GPS locations of the 1000 genomes and what are the alleles that we found, we can create these maps of allele density, those alleles that have been identified to increase survival under drought. And the take home of these experiments was that we typically found drought response alleles in the edges of the geographic distribution. So remember that the North Africa was the warm edge of Arabidopsis native distribution, along with Siberia and North Scandinavia. And this might reflect that there are similar pressures related to water use efficiency at both edges of the distribution. And those are pictures of some plants from those locations. Now, those are maps of droughts responsive alleles, but those might not always be positive or might not always be adaptive in any environment, right? So many traits trade off, or many ecological strategies trade off between environments. So the next question was, can we see in what environments each mutation are selected? So, and this used the two common garden experiments that we had done in Spain, in the edge of the distribution, and Germany in the center. And this is a plot which is kind of complicated, but very telling. Here we have 1 dot, 1 allele in the genome. They are placed in these scatter plots based on where those alleles have been found geographically in the distribution of Arabidopsis thaliana, and then extracting bioclimatic variables. So these are alleles that have been found only in populations that come from high annual precipitation and low seasonality. And then, and vice versa in the other side. And then I color them based on if they increase fitness and survival in a Spain condition, which has low rainfall, or they decrease fitness. And what we see is that alleles that actually are present in populations that come from low precipitation environments appear to increase fitness in this Spanish experiment. And the further they come from higher precipitation environment, the more negatively they impact fitness, probably revealing these trade-offs. What this, in other words, tells us is that the genotypes that are local are likely the ones that have the highest fitness, and the farther we plant, or the farther we bring genotypes from, the less fit they are. And this is a signal of local adaptation at the genetic level. And we can do the reverse with another experiment, which we did in Germany, which is a high precipitation environment. And we can color the same mutations based on whether they increased or decreased fitness in Germany. And again, things that typically come from high precipitation environments were more selected in Germany, green, versus those that come from low precipitation environments, showing this trade-off in fitness, meaning there's not a specific genotype combination that will be most successful in all the environments that a species encounters. And there will be a perfect or ideal mismatch between certain genotype and each environment encountered. So we can use these concepts of how far an allele comes to, and how fit it is to an environment, to try to understand a selective pressure from future climate changes. So what I did in this computational experiment was downloading climate data, 100 bioclimatic variables from WorldClim in the present, and in 2050 from Intergovernmental Panel for Climate Change Predictions. I subtracted them and I made predictions based on these maps on what are the, how far the environment is going to change and how far the alleles at each location are going to be from the perfect match, given the current allele climate matches. What this reveals is, in more simple terms, is that for all these populations in red, these populations have alleles that will become more negatively selected. Climate is changing in a direction towards creating maladaptation, versus populations in white or in green contain alleles that are predicted to be in a similar local adaptation equilibrium. So you can imagine that if we had this type of maps for a number of threatened species, we could try to do more genomic informed conservation decisions on where there are more vulnerable populations given their genetic makeup. All right, so using Arabidopsis, I've shown how we can use genome to phenotype approaches in common gardens to discover genetic variation that could make some populations adapt to certain climates, and how we can start thinking of computational models that could enable differential vulnerability maps of species. So the final question is, well, there might be genetic variation in a species to adapt to climates, but sometimes those variants might be very uncommon and might not become frequent enough to enable a population, for example, to reach a higher, fitness. So how fast can evolution happen from pre-existing genetic variation? In order to ask this, we built a global network of experimenters to plant the same rich mixture of Arabidopsis ecotype from over 200 native populations. So we sent participants from about 45 locations in the world, where you see all these green dots, bags of seeds, and some experimental tools to set up 12 subpopulations at each location and just let them be. And this started in 2017. Every year, when populations grow and reproduce, we ask participants to sample several hundred flowers so that we can sequence their genomes. The idea is that with this sequencing, which we do in pools of flowers, we might identify genotypes that become more and more enriched in different environments. And along with that, we will have demographic and census data to also tell us, did these populations survive or did they die? All right, so some technical areas on how we can really do this. This was a huge endeavor that was conducted by a research assistant in our laboratory that set up new library protocols to sequence multiple individuals at a time and 96 populations at a time. And I'm just putting this glamour shot here to acknowledge all that huge effort. And this is a data set that Rue has created and finalized, and we just have in our server just from the last couple of months. And we're starting to analyze that. But how do we analyze this? It's a little bit more complicated than having each individual sequence separately. So the concept is that when we sequence pools of flowers, we might not know from what individual each DNA read comes from. But what we can do is we can map it to our reference genome, which is the string of letters, and then see at specific positions DNA pieces that are variable. And from the ratios of the different bases that we encounter, we can calculate frequencies. Now, if we have some reference frequencies, for example, by sequencing the seeds, which we've also done, we can compare founder and derived evolved populations and try to infer something from the allofrequency trajectories. So we're expecting, under evolutionary theory, that stochastic allofrequency trajectories will be due to genetic drift, due to the fact that populations have finite numbers, so allofrequencies fluctuate. But if we encounter deterministic trajectories, especially if these trajectories are shared across multiple environments, then we can infer something about natural selection. Those genotypes might have provided some type of adaptive advantage, and that's why they become more prevalent. And if that happens efficient enough, those might be the ones causing that a population is maintained or persist in a location. And this is not trivial, but we've started to invest quite a lot in creating new software, fast software that outperforms current tools and that enables to manipulate frequencies along the genomes for many populations rather than individual ACTGs, which is what most software typically created for human genetics does. All right, so what have we found so far? And these are very fresh preliminary analysis. First of all, this is a plot of all the locations where we have stations in colors. The majority of stations actually survive populations established for at least 1 year, which are in green, many of which survive for multiple years. In some stations, typically in very cold places, they died, but they established but never reproduced. And we have a few anecdotes, such as in UK, of infestations of slugs that destroyed our experiments. So they logistically failed. So these are all the samples that we have, over 2,000 samples, and we nicely see that the majority of adults, of course, are in the spring, which is when Arabidopsis reproduces, although some locations also enable a second generation. And this is interesting because one of the first times that it's actually shown at large scale. So you can imagine that this will be an amazing demographic data set to just do life history models and understand and parameterize where and when Arabidopsis might be living. And that demographic information is something that will be very important to make future predictions. Right, so as I was saying, the populations from actually colder environments are the ones that suffer the most. And interestingly, populations from high temperature and low precipitation survive at least from some for a generation, such as the population in Israel that is beyond the warm edge. So that's actually super interesting, and we cannot wait to see what genotypes those have. All right, so the first look at the data, and this was just 10% of the data that we created of the 280 populations, which encompass about 10,000 seeds, 10,000 flowers from all the locations in the first year. So this is kind of rough general trends of allofrequencies. Was that if we look at this map of or this scatter plot of the starting frequency of all the alleles in the genome from the founder seeds, and look at how much allofrequency changes, which can be maximum 100% change if they started at very low frequency, right? Or minus 100% change if they started very prevalently. What we find is that there is actually a pretty fast change of about 5% average allele frequency along the genome in a single generation. And that goes along with recent estimates from colleagues in the Drosophila melanogaster fruit fly genomics field, and some experiments with another Brassicaceae plant. We also find that it looks like there is a bias. Alleles that are referenced but are less common become more common. And this could be explained, or there are at least some theoretical work that explain potentially rare alleles are those that might be used to adapt in new environments. So that could fit that trend. And finally, because we're expecting the majority of allofrequency changes to actually be just because of co-occurrence of a mutation, neutral mutation with a causal adaptive one, we studied the signal for synonymous mutations and non-synonymous mutations. Non-synonymous are those mutations that change an amino acid. So protein structure is changed, and we expect them to typically, have some type of effect in an organism, versus synonymous, which, although we know that could have some potential effect, they're typically considered neutral baseline. And we see that those mutations that actually change protein, they are evolving, that is, they're changing in frequency faster. So those are a lot of suggestive, interesting preliminary analysis. And we are right now trying to understand these trends and per environment. So we're repeating this for each location where we're plants survived. But just to give you a teaser, before GreenNet, we did a pilot experiment in order to try to see if our methods will make sense, which was important. And this was an example of the same or almost the same founder seeds with 3 subpopulations where we sequence them over a generation. And this is a genome plot on allele frequency trajectory or standardized allele frequency trajectory, FST, between the control lines, which was sequencing of the seed bank, and the survivors on a single generation. And this is a zoom in to an area that is a gene called flowering locus C that we know is a gene that is typically under selection in common garden experiments of Arabidopsis. And what this shows is that in multiple of the independent replicates, the surviving plants had almost completely different allele frequencies at a region that is the promoter area of FLC. And they seem to coincide with other independent GWAS, genome-wide association with SeedSet, which we've conducted in other experiments. So this was very interesting, and we'll probably repeat this kind of analysis for each location, identifying regions in the genome that might be responsible for survival of those plants. All right, so with that, I've shown, at least in the last one, preliminary data that changes in allele frequencies when present can actually happen rapid. And some of these do not look stochastic, but they look deterministic in agreement with what might be expected with natural selection, increasing their fitness. So they might be related to adaptation. And with that, I want to thank everybody. Oh, I had one more, but I'll skip it. You can ask me about this in the round of questions, and it's about CRISPR-Cas9. With that, I want to thank everybody in our laboratory, all our funders, long-term supporters, collaborators of the genetic extinction, new research, and all the participants in the GreenNet network that have been working for now almost 5 years in sampling flowers in the field with us. And thank you all for your attention.
Okay, thank you very much. Looks like you've gotten lots of silent Zoom type applause. Yeah, very nice seminar. So I think the way I'd like to handle questions is that if you have a question, don't type the whole thing in a chat box, but put your name in the chat box, and that way I can just